Welcome, Fungophiles. We're glad to have you here tonight. My name is Marisa Gomez. I'm the Public Programs Manager at the Santa Cruz Museum of Natural History, and we are presenting tonight's lecture in partnership with the Kenneth S. Norris Center for Natural History and the San Lorenzo Valley Museum, and we're going to share more about that partnership in just a minute. I also wanted to take a moment to acknowledge that the land from which I am joining you and the land on which the Santa Cruz Museum of Natural History resides is the traditional and unceded territory of the Yupi tribe of the Awaswas Nation. Today, these lands are stewarded by the Amamutsan tribal band, and the Amamutsan are working hard to fulfill their obligation to Creator, to care for and steward Mother Earth and all living things through relearning efforts and the Amamutsan Land Trust. And we would also love to acknowledge the Indigenous stewards of the land where you are. So if you know the name of the tribe whose homeland you are streaming in from, please share that in the chat. And if you're unsure, I actually have a website that I can share with you all here. And you can look up the Indigenous stewards of the land where you are there, and you can add it to the chat. And one last little bit before we get started is I do want to acknowledge again that this program is in partnership with the Kenneth S. Norris Center for Natural History and the San Lorenzo Valley Museum. And that is because of their new exhibit, Look, Act, Inspire, Sustaining and Expanding the Community of Naturalists in Santa Cruz County. And so to speak more about that exhibit and how this program is in support of that exhibit, I want to bring in the director of the Norris Center, Chris Lay. Chris, you're on.
Hi, everybody. Welcome to this talk, and thanks, Marisa. We're excited to have the Santa Cruz Museum of Natural History as a sponsor for our exhibit and to work with you on this speaker series. Welcome, everyone, to this is our first presentation of our program associated with our new exhibit, and we've called it Look, Act, Inspire, Sustaining and Expanding the Community of Naturalists in Santa Cruz County. Fungophiles are definitely part of that community. And it's an exhibit that really is celebrating the large, diverse, and growing community of naturalists in our county. Like Marisa said, I'm the administrative director of the Ken Norris Center for Natural History at UC Santa Cruz. We work with students of all ages to help them develop their skills as naturalists and nourish their individual passion and curiosity for nature. And we do that in many ways. And one way we accomplish this is through collaborating with community organizations and hosting public events like this one. This program is associated with an in-person exhibit, and that in-person exhibit, we've partnered with the San Lorenzo Valley Museum, and we're excited to be one of the first exhibitors in their new Belardi Gallery in Felton. And as many San Lorenzo Valley residents know, that's in the same building as the old Felton Library. And we are hopeful that we are on the road towards opening that museum. We actually have set up most of the in-person exhibit that's associated with this program. And it's not open yet, obviously, but we are hopeful that it will open perhaps in weeks or maybe months. So please stay in touch with us. Check your email if you're getting our emails from the Norris Center. Look at our website, look at our Facebook page for updates on when you can visit. So fortunately, what we did do, and Marisa just put this up on the screen, is we created a accompanying website that goes along with our exhibit. And you'll find bios of many of the naturalists we interviewed. We interviewed almost 100 people, which is just a subset of the community of naturalists in Santa Cruz County. But that includes Christian Schwarz, who we're going to hear from tonight. It also includes an expanded section about several community naturalist leaders, including Fred Mcpherson, who is a longtime naturalist of the San Lorenzo Valley. And I just want to point out one thing. If Marisa, if you would go to the upcoming events, we have more events, and we have even more planned that aren't on this page yet, but I just want to point you to the next one. we're going to do a special workshop / presentation about Fred Mcpherson and his contributions of over 50 years of natural history observations in the San Lorenzo Valley and his evolving perspective, starting as a nature journaler and moving into videography. And so I encourage you to check that out. You can register right from this page to come to that talk, and it'll be a participatory event as well. So check that out. And yeah, keep checking back. We hope we can open the in-person exhibit so that you can come up and see the actual exhibits that we've set up. So thank you for coming, and I'll turn it back over to Marisa.
Yeah, thanks, Chris. And I can't wait to see the physical version. We're in a similar boat over at the Santa Cruz Museum of Natural History, where we've got lots that we want to show off in person, but we're lucky to be able to do so in the the digital realm while that's where we are. So let me pull back up one other slide to share. And I just wanted to, last thing before we welcome in Christian, is point out that this program is also kind of another partnership for the Norris Center and the Santa Cruz Museum of Natural History, because we are also partnering on a new initiative to implement community science to look at the impacts of the CZU Lightning Complex fires on our community. And that's going to launch officially next month. And that is in partnership with the Norris Center. And it's also in partnership with the California Native Plant Society. So this talk with Christian is sort of like a kickoff for that. And on that note, let me chat a little bit about the topic du jour, which is mushrooms with Christian. So I'm going to be welcoming Christian. And first, I just want to say a little about him, let you know who he is. So Christian Schwarz is a dear friend to both the Santa Cruz Museum of Natural History and the Norris Center. He's done, I think this is his 5th program with the Museum of Natural History in the past year. So we're very grateful for that. And he's a research associate at the Norris Center. He studied Ecology and Evolution at UC Santa Cruz and now spends his time photographing, teaching about, collecting, and researching macrofungi. And he's co-author of Mushrooms of the Redwood Coast along with Noah Siegel. And just an all-around amazing resource for our local community. But really, like the broader community too, the world. We're lucky to have him so close, but also realize that his work impacts people far and wide. I'm sure there are people joining us today who are not from the direct Santa Cruz area who appreciate his insights. And he's an enthusiastic and inspiring educator. And so now I'm going to invite him in to inspire and educate. So Christian, if you want to come on back.
Hi, everybody.
Take it away.
Alrighty, folks. Well, thanks to Marisa and to Chris and to the rest of you who helped put this on. I'm glad to be talking about fire, because clearly everyone who lives in Santa Cruz and the extended Santa Cruz County, San Mateo, Santa Cruz Mountains community has been pretty either directly or indirectly affected by fire in the past year. The CZU fire happened not quite a year ago yet, so it's still in very recent memory, and clearly the effects of the CZU fire are going to continue to impact our community for a long time to come. So I'm talking not just about fire, but about sort of the long-term monitoring project that I have done in the Santa Cruz Mountains, centered on macrofungi for the past few years, and specifically winding up looking at the impacts of fire on that monitoring effort. So I'm gonna get right into it here, but yeah, the first part of the talk is less specifically about fire, but towards the end I will talk directly about the impacts that fire has had on the monitoring transects that I've been looking at. So here we go. I think my screen is now shared. Can I get a confirmation on that, Marisa or Chris?
Confirmed.
Okay, great. And here we go. So I'll just start by defining what long-term macrofungi monitoring even is. And one way of talking about this is by contrast to what I normally did before I had started this project, I would just sort of go wherever I felt like on any given day to look for mushrooms. I would go to my favorite spot, and then I would go to another favorite spot, and then I would explore a new place that I'd never been before. And sometimes that wouldn't pan out, or it would become a new favorite. But it was very haphazard. I would scatter my effort and my time all around the county, sort of as I felt like it. Sometimes I'd go to the same place 2 days in a row, or I'd never visit it in the course of a year. So it was all very unpredictable. And that was really fun and a really good way to get myself up to speed with the diversity of fungi that occur in the county. And it was the best way to sort of get photos of a lot of the species that live here. But after about a decade of doing this, I realized it was likely that I was missing a deeper understanding of the dynamics of these mushrooms, I was probably even missing some of the species that occur for very brief windows of time. And I started to have a desire to make my efforts more structured, more standardized, so that I was visiting the same locations repeatedly, building sort of a more intimate familiarity with a few locations rather than scattering my efforts over the whole county or the whole Santa Cruz Mountains area. So long-term macrofungal monitoring, to define this, is basically surveying the same locations, however you define that, over a multi-year or even multi-decadal duration. There are examples of studies like this happening in Europe that have persisted decade after decade. But here in the United States, studies like this are really scarce. There's very few data sets where people followed up on mushroom dynamics in this sort of repeated way, even for a few years. So my goal was just to do it for 3 years. And I'll talk more about why 3 later. But the idea is to do this frequently. So in my case, I was doing this every 7 to 10 days or so, somewhere between a week and 2 weeks, somewhere in that window, and to do it regularly. So to keep this up through the course of the mushroom season, which broadly defined in our area is from late October into early May. Not only that, I was not just going to be monitoring sort of open-endedly, but trying to quantify area and time. And when you quantify area and time, so how much time you spend and how much area you cover, this is what ecologists call effort. So we standardized I standardized my effort. And this is important for comparisons in the future because people can spend the same amount of time and search the same amount of area, and then you can divide by that effort to sort of determine whether or not there have been actual real changes in the commonness or rarity of a particular species. So I tried to establish a protocol for myself that would be easy to follow. I would establish some 1 kilometer long transects, and I'll talk about what a transect is in a second here, and I would walk them from the start to the end and then backwards, because I think any of you who are mushroom hunters have experienced that when you walk a trail in one direction, you always see different things when you walk backwards and see it from the other perspective. So I wanted to include both of those perspectives, and I would sort of split my time between the out and the back. I would record my start time and my end time, so it'd be easy to calculate how much time I spent later on. And I would record every single species that I encountered on that transect in the time that I spent there. Not only that, but for each species that I found, I would record the abundance of those species. So how many of them I found. And that would sort of help me differentiate between a year where I only saw 1 fruit body and a year where I saw 100 or 1,000 of that same species. So here's a map of the fungi that I've seen in Santa Cruz County from iNaturalist. And you'll notice that in the past 4 years, which is or 3 years, which is what's represented here, there are big clusters of observations of mine. And that's not a coincidence. That's because of these 10 transects that I established around the county. So I've been visiting these over and over repeatedly for the past 3 years. And that's why there's big clusters of observations here. Here's a map of the transects themselves. So these are not all to scale, obviously, but there are 10 of them, and they're scattered around the county in such a way that I have situated them all mostly in redwood forests. So they're all sort of the same kind of habitat type. They're all 1 kilometer long, and many of them are in pairs or triads. So they allow for sort of replicates of similar environmental conditions and similar forest types. But they range all the way up to Big Basin, my highest elevation transects, all the way down Ben Lomond Mountain to the mid elevations of Marshall Field and Wilder Ranch, down to Santa Cruz campus, the UC Santa Cruz campus. And then I have 2 transects out at Nicene Marks, which aren't on Ben Lomond Mountain. So a slightly different pair of transects there. Okay, so around each of these trails, how was I going to record fungi? Which mushrooms would I decide to record in my notebook? The way I thought about this was that I would try to establish the center of the trail and 1 meter on either side of it as being the core of my survey route. I would try and record everything within 1 meter of the trail edges. I would try and record most things within 2 meters. And then by the time I got out to 10 meters, I was really not recording mushrooms anymore. So if you take the idea of this curve as being how much time or effort I was spending within a certain distance of the trail, it would look approximately like this. Oops. Right at the center of the trail, I would really try and record every single mushroom that I saw within 1 meter. And then within 2 meters, about 95% of my effort would cover that area. And by the time I got out to 10 meters, I would basically not be looking at mushrooms anymore. I would not record any mushrooms further than 10 meters from the trail. And really, 99.7% of my effort would be within the 3 meters on either side. So these really are trail side surveys. And the reason that I decided to put it together this way is so that it would be easy for people to redo these surveys. You wouldn't have to go stomping through the woods or out finding a trail or finding a transect somewhere inaccessible. All of these trail or these transects are set up in such a way that you can drive to the start of the trail and just park there and get out and start surveying. So my goal was to make the protocol and the transects themselves easy to follow up on so that there would be a chance that there would be some persistence to this data collection over time. So if you take one of these transects and draw one of these curves over it, you can then draw sort of a vague or blobby area around it, such that you can pretty much estimate or quantify the total area that you've covered, and then it is really helpful for quantifying effort in the future. So this is how I established my protocol, and I don't want to spend too much time on this, but people often ask me, how do you decide what to count? And this is what the process that went into it. So I set up 10 transects all around the county, like I said, that I visited every 7 to 10 days between October and May. So over 3 years, this adds up to about 800 kilometers of walking, 500 hours of direct survey time. And at this point, it's shaping up to be about 11,000 individual records of mushroom diversity and abundance put together, associated with the metadata on these transects. So it's a lot of data to deal with. And in retrospect, I wish I hadn't done quite so many transects. This was a little bit more than I could, a bit off more than I could chew. I would have either shortened the transects or done fewer of them, but that's how it went. So I really don't encourage anyone to take on 10 transects all at once. I think it's a much more feasible scale for people who follow up with this protocol to do just 1 or 2 or a couple. And here's what my notebook looks like on an average day of surveying. Actually, this is not an average day. This was kind of a good day. This is on the UC Santa Cruz Upper Campus Transect. And you can see there was something like 91, 92 species of fungi that I encountered on my transect, as well as numbers indicating how many of each of those species I found. So eventually I go back and I digitize all these records and then I do data analysis, but that's later. A lot of that data analysis, the number crunching, is yet to come. So what did I find out? After the first year, it was pretty clear that even a 1 kilometer transect contains a lot of diversity, pretty much no matter where you place it in Redwood Forest in Santa Cruz County. Each one of these transects had more than 330 species, and that's pretty incredible. If you think about a 1 kilometer transect, you can walk that, pretty quickly in 30 minutes. And pretty much no matter which 1 kilometer chunk of the county you decide on, a very narrow trail side survey, you will have a good chance of encountering more than 300 species of mushrooms in that tiny little area. But comparing the least diverse transect to the most diverse transect, there was 100 species difference. So the most diverse transects were 100 species more diverse than the least diverse transects. So the potential for important differences between habitats to translate into diversity differences was real. There was a lot more diversity in some places than others. A full, quarter more, or a third more. The least diverse transects, not surprisingly, were those that were most disturbed or urbanized or impacted by human activity, whereas the most diverse transects were the ones that were sort of more old growth or more mixed forest types. So there was a big range, and I actually have found more species since the first year, so I have to update these numbers eventually. And what this looks like, it's hard to communicate, but on some of, or if you add all of these transects together, there's easily more than 550 species recorded now, just on this tiny picture of the county that's painted by 10 1 kilometer transects. There's over 550 species, which suggests that if we keep doing this kind of surveying more broadly in the county, we're going to have thousands of species of macrofungi that call our county home, and potentially something like 3,000 or 4,000 once all is said and done. And of course, all is never said and done when it comes to surveying for biodiversity. You'll always find new things, even decades down the road. So there's a lot to be learned about the diversity of fungi in our tiny little county. So I'm just going to briefly show you some of the sort of most exciting things, the things that people get worked up about: the disjunct, the rare, and the undescribed. So these are species that, in the case of disjunct species, means that they are populations that are far away from the core of their range. Rare species are species that obviously are just globally rare. They're hard to find no matter where you are. And then undescribed species are species that don't have names yet, that are new to science and need someone to name them, which is always exciting. So here's just a few examples. Here's a disjunct species called Hayderia. It's this really adorable little beige matchstick that grows on Douglas fir needles. And this is a species that's much more common up in Northern California and in the Sierra Nevada in the mountains, growing on fir needles, but it's quite rare this far south, and it's pretty rare on the coast. But Santa Cruz County appears to be one of the southernmost places you can find this species. So this is a good example of an exciting disjunct fungal taxon. Hayderia aviatus. Here's an example of a really rare species. Now, the name of the species is Lepidorodophila, and until I found these individuals, it was only known from one place in the entire world. There was only one place that was known to find this species, which is where it was originally described, in a cypress forest in San Mateo County. And it was thought to be critically endangered, and actually still is. But at least we know now that there's at least one other place where this mushroom occurs in the world, and that's right here in Santa Cruz County. This mycelium is at Nicene Marks, growing under redwood. So I was really excited to find this species because of its rarity. And it is a species I don't think I would have found had I not had this very structured, rigorous 7 to 10 day interval, because this is a mushroom that comes up and goes away, disappears within a 7-day window. So it's a very short-lived species, and you can easily miss it if you happen not to be in the right place at the right time during its brief window of fruiting. So this is something that I credit the more structured protocol with finding. This is a pretty drab little mushroom, but what's really exciting about it is that this is a genus that is very poorly known in North America. There's basically no records of it anywhere. And it was only because I was forcing myself to make sure that I identified every mushroom on my transects as best I could that I stopped to pause and take the time to really, truly try and figure out what this thing was. And I brought specimens of it back to my natural history of fungi class last year that I'm teaching at UC Santa Cruz, and the undergraduate students there included it in their sequencing, their DNA sequencing lab that we did with Christoph Teige at the Hausler lab. And we found out that genetically, there was evidence to put it in this genus Mycinella, which is one of the few records of this group of mushrooms from anywhere in California and really anywhere in North America. So it's really special to find, some of the first evidence of a whole group of organisms right here in our home county. Another one that's very similar to that story is this Tricosapi, kind of a drab mushroom, but it's got really cute little hairs all over the cap. That's actually what the Latin name means. Trico is hair and Sabi is head. So this is a little hair head. And it's actually common, present on a number of my transects, but it doesn't appear to have a scientific name. And once again, it was the first record of this whole genus of mushrooms. No close relatives were known from California until we started finding this species here. So Santa Cruz leads the way. So those are just some examples of the exciting sort of biodiversity, the species level biodiversity. But what I was much more curious about, personally, much more interested in finding out, is what patterns in time and space would emerge from the data that I was collecting. How long do fruit bodies last? What is the shape of the progression of a mushroom season in a given year? And so now I'm going to tell you a little bit about these questions of time and space. This is a really gorgeous mushroom, very subtle pastel colors that has sort of blue-green, powder blue tones, yellow, chartreuse. There's even some violet colors down at the base of the stem. This is a mushroom called Gymnopolis Punctifolius, and it's associated with old-growth conifer forests. So in our areas, this is with old-growth redwood, old-growth Douglas fir. And it's really not common in Santa Cruz County at all. There's only like 2 or 3 locations where it's been found in the county. I found one fruit body of this on one of my transects the very first year that I was doing this. And I was excited because it's a rare mushroom in our area. And I took special note of it because it was only one. And every time I revisited that transect, I made sure to look for it. And it turned out that that single fruit body lasted a really long time. And week after week, month after month, I was able to find this individual still in pretty fresh, good condition, still seemed like it was actively growing and releasing spores. I actually have data that indicate that it was present for at least 105 days. So it was just a really persistent mushroom fruit body. This is not something that I understood about mushrooms. I didn't have any sense that they could last anywhere near this long. So that was the kind of pattern that I was looking for when I started this project, is to get a real natural history understanding of the life cycle of these organisms. Another pattern that emerged that was totally unsurprising, this is exactly what you'd expect if you're a mushroom hunter, there is such a thing as a mushroom season. It's a real pattern. In October, when it's dry, there's not many mushrooms. In the middle of the winter, when it's really wet, there's a lot of mushrooms. And then by the time you get to April, where it's sort of drying out and warming up again, there's not many mushrooms again. So it makes this sort of, generally speaking, bell curve of diversity and abundance throughout the season. So this is the plot just from the first year of data that I took. And you can see that there's at least a couple transects where it gets really, really diverse in the middle of the mushroom season, in the middle of the wet season. What I'm really looking forward to being able to do, though, is to take a graph like this from one year and a graph like this from the next year and compare them to show how the bell curve shifts. And in some years, we expect it to not look like a single peak bell curve, but perhaps have two peaks, or have a peak that's really lopsided over on one end of the season. If it's a late winter or a really good wet spring, we would expect that the tails and the distributions look a little bit different. And then if you do that on a decade to decade basis, you can start to compare much longer term patterns in a sort of rigorous data supported way, which I'm really excited to see what people do with this. So those are questions of time and space, and now I want to get even further out there and talk about other elements, water, earth, wind, and fire, and how these elements influence fungi. So water and earth are sort of the most obvious in their impacts that they have on fungal diversity. So rainfall, which is how water comes into this, is the single greatest determinant of diversity and abundance of mushrooms on any given survey. And that's not a surprise to anyone who's been a mushroom hunter. If it's not raining, or if it hasn't rained recently, you're not going to find many mushrooms. If it's been raining a lot, you're much more likely to find a good diversity of species and abundance of species. But you also have to factor in questions about soil. And what I mean about earth here is really more habitat in general. What are the habitat conditions that the fungi live in? Because you can't just go to the middle of downtown after a rainstorm and expect to find mushrooms. You want to be in the forest if you want to find a good diversity and a good abundance of fungi. So the combination of habitat and rainfall, so water and earth, so to speak, is these two factors together are the main drivers of how many species you'll find and how many fruit bodies, what abundance of those species you'll find. But this is predictable. I sort of already knew this intuitively. What I did not have such a good idea about, or such a clear picture of, was the impacts of wind and fire, for obvious reasons. One is that wind is sort of subtle. If you're not visiting the same places very frequently, the impacts of wind can be easy to miss. And fire, obviously, I didn't have any impact or any experience with because it hadn't impacted my home forests. I'd never had a close-up view of a wildfire in Santa Cruz County. So I'm going to talk about those now. So I was really surprised to learn. Well, I wasn't so much surprised. I was. It made a lot of sense once I encountered it. It makes complete sort of logical sense that wind should be a really big influence on the diversity and abundance of mushrooms, but I hadn't ever sort of directly experienced that before. So the reason wind has such a big influence on mushroom diversity and abundance is that it is a major drying force. Wind sort of pulls water out of mushrooms and pulls water out of soil. So it's sort of the anti-rain. It does the opposite of what rain does. It dries out soil even faster than warm temperatures. A few days of warmth is not going to dry out the soil as much as one day of strong wind. So I think that my data will show, although I didn't really quantify wind because I was not able to, I didn't think ahead of time to set up a way of measuring wind, air currents, airflow in my transects. But I think my data will show that there are dramatic changes that are more pronounced in mushrooms that have small fruit bodies on short time scales. So what do I mean by this? Well, here's just a picture of some of these mycena that oftentimes you can find in carpets of thousands of mushrooms in the understory of a conifer forest, where each individual mushroom is smaller than your pinky, but there might be hundreds or thousands of them. And this is the kind of mushroom where one day you might go into the woods and see thousands, and a day later or 2 days later, they might almost all be gone, because these are extremely susceptible to drying by wind. And I think this means that wind is a major evolutionary selective pressure on mushroom fruit body morphology. So this is what these mushrooms look like up close. And any mushroom that looks like this, that's this small and delicate, is a mushroom that has to worry, evolutionarily speaking, about wind. Natural selection is going to operate in response to wind. Here's another example of a mushroom that has reduced its fruit body down to the bare minimum. It's really just a slender thread, a noodle of fertile tissue. And these mushrooms dry out instantly, like that, if there's any wind whatsoever. So this is the kind of mushroom whose fruiting triggers, when it decides to produce its reproductive structures, is going to be really closely timed in relation to wind. And how you predict that, I mean, obviously these are not the terms that we want to use when we're talking about evolutionary adaptations, but how that sort of response is coded into their biology is something that I think deserves a lot of investigation. Okay, now I'm going to talk about fire. So this is a picture I took just a couple days after the CZU fire started, when the fire and the smoke was bearing down on Swanton Road, coming downhill towards the coast. These are Monterey pines, Monterey cypresses that you see in this picture. A lot of smoke in the air, which is why it looks so orange. And in this place where we were standing when I took this photo, it eventually burned all the way to the beach sand. So it burned all the way down this slope and even burned ice plant, which is a really hard plant to burn. It's basically pure water. But the fire was so intense there that it drove all the way down the hill to the ocean, burning vegetation literally within a stone's throw of the waves. So the CZU fire was in the late summer of 2020. It in total burned something like 86,000 acres, if I have my figures right, and almost all of that area burned at high intensity. So this was not a creeping ground fire. This was not the so-called good fires of controlled burns or understory fires that sort of clean out the choked vegetation of denser forests. This was a crown fire in many places, where the flames reached all the way up into the canopies of the trees and probably caused significant mortality of a lot of trees. Almost all of Ben Lomond Mountain was burned. That's where the fire was centered, starting in Big Basin and going outwards from there. And about 25% of our county's land area was burned, which is just an astonishing statistic. And then the forest started. It's worth noting that the main habitat type that was impacted is mixed evergreen forest, including and especially the old growth redwoods of Big Basin. So here's some satellite views, some aerial views of the before and after of Ben Lomond Mountain before the CZU fire and after the CZU fire. And you can see just how extensive the fire scar is, just how much area was impacted or affected by this event. A lot of the upper elevations of Big Basin look like this in the immediate aftermath of the fire. There's a thick layer of ash on the ground, but nothing else. There's no remaining logs, there's no remaining duff, there's no remaining twigs or leaf litter. All of it was converted to carbon. It was carbonized, turned to soft, loose ash. And you can see that the branches of the trees are defoliated. You can't even see the tops of the trees in this photo, and none of them have any leaves left. It burned every leaf off to a height of tens of meters. Here's a deeper view into Big Basin shortly after the fire. And you can see, once again, there's a few logs here because they're larger diameter and a few branches, but those likely fell after the fire ended. It was probably a little bit lower intensity here because you can see the tan oak trees still have some of their leaves on, but really extensive burns. Something that I have yet to actually understand is why tan oak trees burn differently. You'll notice that this slender tree in the middle of the image, the bark is completely unblackened, even though the very large Douglas fir on the right and the big redwood on the left, which are right next to it, have very severely carbonized blackened bark all the way up the trunk. So something about tan oak bark is very different in how it's affected by being directly exposed to flames. And this is a fascinating question that I'm not even going to begin to investigate. Someone else has to do. Maybe they already have, and I just don't know it. Redwoods, interestingly, have this ability to do what's called epicormic sprouting. So a lot of the redwood trees in the habitat, the transects that I was surveying, look like this right now. They have just an incredible vital life force, which allows them to resprout from their burned bark pretty instantly, pretty much immediately, which is not the case for most of the trees. Redwoods are sort of especially resilient to this kind of impact. Here's an example of redwoods that are sort of resprouting really vigorously, especially at their base as well as on their upper trunks. The tan oak tree at the right is only resprouting around the base, and I'll talk a little bit more about that in a second. So these were the diversity estimates of all the different mushrooms on my transects before the fire. So there was a range from about 330 species over on the left to about 430 species in the most diverse transect on the right. And 4 of these were burned in the CZU fire, including some of the most diverse transects, which was kind of hard to handle. It was pretty tragic to think about when the fire was happening, that some of my richest and most wonderful transects that I was really excited to go survey the diversity of were burning. But in a way, I ended up having to take it as an opportunity to more closely study this fire system. So I had to try and take it as, for what it was worth, to try and take it as an opportunity to study fire. And something that has become clear to me is that all organisms, not just fungi, have a range of responses to fire. There are those that we can categorize as fire sensitive / fire intolerant. So these are species that really don't do well with exposure to fire. They die or they disappear from an area after it burns. There are fire tolerant species, which sort of can put up with a fire, but they're not necessarily benefiting from it, but they can get through it and they're somewhat resilient. There are those that are fire stimulated, so they actually have an increase in their vigor or their growth or their reproduction after a fire, after they're exposed to flame. And then there are those that we can call fire dependent, which are actually, they need fire. It's a necessary precursor to their reproduction. They won't actually be present in a habitat, at least in any sort of non-dormant way, until that fire occurs. So just as some plant examples, a fire intolerant species is something like a Douglas fir. And if you look over at the upper right, this burning, actively burning, hollowed out Douglas fir stump in the upper right, that was burning 4 months after the fire originally started. So this Douglas fir continued to smolder, and Douglas firs are an example of a tree that really can't tolerate fire. They don't resprout. They go up in flames pretty much instantly, all the way to their canopy, to their crown. And so it's thought that 80% or more of the Douglas fir in Big Basin may have just outright died in this fire event. A fire sensitive species might be something like a tan oak, where they do resprout really vigorously. That's at the lower left of the images here.
Sure.
So the lower left of this image is a tan oak tree that is resprouting from its base, but it lost all its leaves higher up into the canopy. So it's what I would call sensitive, because although it survived, it's going to have a really hard time reaching its former sort of stature and capacity. It's not going to photosynthesize anywhere near as much. And it's basically been reduced to a low shrub. This tree might have been 30 or 40 meters tall, and now it's just 2 or 3 meters tall. So it's a sensitive species. They can't sort of regrow from their trunk the way a redwood can. As an example of a fire-stimulated species, that might be something like a Mariposa lily, which you see in the middle right of this image. These are bulb plants. They have an underground bulb that lasts from year to year. And so that bulb is very insulated from fire. But for whatever reason, the chemical changes associated with the disturbance of fire cause Mariposa lilies, of some species anyways, to have really vigorous production of flowers in the years following. But they'll be there even if there isn't fire. So they don't need fire, but they're stimulated by it. Down at the bottom right, oops, this is supposed to say Santa Cruz Mountains beard tongue, not just Santa Cruz, but that is an example of a plant locally that seems to really need fire to be common. It is a rare species locally, most years, but in years following a fire, or rather in the months following a fire, really in the near term, they bloom in abundance. So this Santa Cruz Mountains beard tongue, which is a kind of penstemon for those of you who are curious, just really ramps up its flowering, its seed release. So it is something that does appear to be a fire follower. So what does this look like, not in the world of plants, but in fungi? Oh, there it is, Santa Cruz Mountain beard tongue. That's what was supposed to show up. So in fungi, we don't really know what fire intolerant species look like. We don't really know what fire sensitive species are. I'm starting to build a guess based on the data I'm collecting that some kinds of agaricus mushrooms, like this is the same genus of mushrooms as the portobello mushrooms that you have in your kitchen, the button mushrooms, these appear to be pretty robust, kind of scrappy, resilient to fire disturbance. They were the first group of fleshy mushrooms that I found in Big Basin after the fire, but there weren't many of them. So they don't really appear to be stimulated by fire. They didn't love it. They were just hanging on. And then this year, I found out that there's this mushroom that many of you have probably seen in our local forests called the redwood rooter. And I was amazed to learn or to find that there's evidence that it's fire stimulated. And I'll talk about that in more detail in just a second here. But actually, it's sort of in reverse order. Our knowledge of mushrooms is such that we actually know a lot about the fire following species. Things like Anthraco, morels are famously fire followers. But we don't know so much about the species that can't handle fire, at least on the coast. And this is partially because we don't have good, robust, quantified estimates of diversity and abundance. And if you don't have estimates of how common a mushroom is before a fire, it's really hard to compare it to what happens after a fire. So I'm hoping that the data I'm collecting will help to fill in these sort of blank spots about what mushrooms are particularly sensitive or even downright intolerant of fire disturbance. So here's an image of the mushroom I was just mentioning, Coloriza umbonata. It's this somewhat drab-looking mushroom. It's not extremely colorful. It's got no particularly strange appearance, other than that it has a very, very long rooting stem. So this sort of tail tapers down into the duff of redwood forests. And it's fairly common. In fact, it can even be quite common in redwood forests in the fall and the winter, or actually more in the winter and the spring than the fall. So it's there. It's sort of always present in redwood forests of sufficient age and complexity. But what I was amazed to find is that, so for example, on one of these old growth redwood transects, in the 2 years leading up to the fire, I recorded this mushroom 6 times on this particular transect. So, not that many times. And the counts of abundance of this mushroom ranged from 1 to 7. So not too many of them. They were there in low numbers. That is sort of the baseline of how abundant this mushroom was on this transect leading up to the fire. After the fire, on that same transect, which burned quite heavily, I found 125 fruit bodies on my first encounter with it after the fire. And on my second encounter, I found 493 fruit bodies of this mushroom. So it appears to be bona fide stimulated to a much greater abundance than it was ever present beforehand. This is, a massive, many-fold increase in how abundant this mushroom was. So here's an image of just how abundant that looked. Here's a picture of a fairy ring of this mushroom in the Big Basin burn zone after the fire. Anthraco is an example of a mushroom that's a classic fire follower. So this one's actually dependent on fire to reproduce. You almost never see this mushroom in the forest until a fire happens, and then it literally blankets the landscape. There are tens of thousands of these little tiny orange cups covering the charcoal and the carbonized organic matter in Big Basin and Fall Creek and Bonnie Dune. And this is a well-known species. Anthraco, the beginning of this mushroom's genus name, actually means carbon or charcoal. So it's named after this substrate, this habitat preference of post-fire landscapes. Another mushroom that I didn't even know, I'd never really clued into its identity before the fire, is Lentoporia carbonica, this sort of amorphous white polypore that appears to really love burned Douglas fir and redwood trees. And it's one of the most common mushrooms that's out there in the forest right now, at least on the wood anyways. It appears to have been dramatically stimulated by the fire. Whether or not it persists after the fire will help us figure out whether or not it's categorized, we can think of it better as a fire follower or something that was stimulated by the fire. Since I hadn't seen it in these habitats before the fire, I suspect it's a bona fide fire follower. It's actually something that we won't expect to see persist very long after the influence of fire sort of fades away. And then there's brand new discoveries, things that I simply don't know what they are. This mushroom really blew my mind. It is probably one of the most exciting mushrooms I've ever found, not because it's the brightest colored mushroom in the world, not because it's particularly interesting in its structure, although it is kind of an attractive species forming these large clumps. The reason I was so astonished to find this mushroom is that it was one of the first fleshy mushrooms, one of the first gilled mushrooms I found in the burned zones. And I have no idea what it is whatsoever. It doesn't line up with any sort of genus level, family level, species level thing that I know. And it wasn't just present in small numbers. It was present in the tens of thousands. There were so many of these fruit bodies. It was like being surrounded by a UFO the first day that I found these. I really was taken aback. So I collected a bunch of them. I sent them off for DNA sequencing, and if it hadn't been for the deep freeze that's affecting the middle part of the country, delaying mail, I'd probably have the DNA sequence of this mushroom this week. Instead, it'll probably be delayed until next week, but I have confirmation that the PCR products of this mushroom have been sent to the DNA sequencing lab, and we should very soon have a much better idea of what this mysterious fire-following bizarre creature is. I'm so excited to find out. I can't wait. And here's just a few more views of it. I've got a common name that I've been using to refer to it until we figure out what its relationships are. The exuberant ash or cinder cap, because it's so exuberant. It fruits in tens of thousands, giant clusters, troops all over the landscape. And it really is a cinder cap. It often lifts up piles of ash or burned soil, totally gray and powdery on top of its fruit bodies. So exuberant cinder cap, keep an eye out for it. It's out there in the burn zones. It's hard to see exactly how many there are in this image, but hopefully you can see closely enough to see that they are dominating this area. There's more mushroom than there is dirt here. I counted actually every single mushroom in this image, one by one. And this is an area that covers about 3 square meters. So think about the size of a kitchen table or less. And there's something like 293 fruit bodies present in this image. So they just absolutely densely blanket the landscape in some of these burn zones. So they're really not ecologically trivial. This isn't a one here, one there kind of occasional species. Whatever this mushroom is doing, I had never seen it before. It is very closely tied to fire, and it probably plays a big role in the post-fire succession colonization of new species into the burned landscape. So these mushrooms rot, they attract insects, the insects attract birds. The birds are defecating plant seeds that they've eaten, which helps to sort of re-inoculate the area with new plant life. So they're definitely in a post-fire pioneer or recolonization ecological role. So it's really important for us to figure out what these are. We need to be able to communicate about them, which means we need a name for them. And that's sort of where the fun taxonomic part comes in, is describing a new species that's ecologically critical. And I really want to thank the Save the Redwoods League, who took a major chance on funding non-directly plant-related research in redwood habitats. They supported 3 years of these surveys with a grant that I applied for back in 2017. And now I'm coming up on the end of my 3-year survey period, at which point I'll be crunching all the numbers, doing a lot of data analysis, and trying to pull out the most interesting patterns of mushroom diversity and abundance in space and time throughout the habitats of redwood forest in Santa Cruz County. So that's the end of my talk, and I just want to say thanks again to Marisa and Chris and the museum for having me tonight, and I can take questions from you all now.
Thank you so much, Christian. Gosh, that's fascinating stuff. I mean, that exuberant cinder cap is just wild. And
You got to see it, didn't you?
I hope that people will ask some questions in the chat while I ask you my first question, which is, so this mushroom that you had not seen in the 3 years that you were closely looking for it, but also probably just like has not appeared for a while. Like, is it that the mycelium was blanketing all of that like broad swaths of land for like decades and living?
Question number one is, what was this mushroom doing before the fire? Was it latent in the soil, resting in some sort of dormant phase? Was it present as spores? Was it in partnership with some sort of plant host that we're not clear about? That's totally unknown. And I actually am not totally sure how to go about answering it.
It's wild. I can't. It's so much to wrap your head around. I hope that you report back to us on what your exciting package of mail reveals so that we can share it out.
You won't be able to get me to stop talking about it. I've already said when that email comes with the sequence data, I'm clearing my schedule for 2 days.
There'll be, like parties in the street.
Right.
Let's see, we do have some questions. So someone is wondering about the Transects, and so this was a 3-year project. You're reaching the close date. So what is going to happen? You've been doing this for 3 years. You're coming to the end. Is someone going to be looking at these? Are we going to be able to continue to see what the results are for post-fire year 2, or what do you think?
That is my sincere hope, that we can get a sort of continuity, a longevity plan together for this data to continue to be collected. For a variety of reasons, I suspect that I won't be the person who will continue to do that, because it's a fairly intense thing in terms of how much time it takes, and it's hard to integrate the rest of your life with this this sort of intensity of surveying. So I suspect that it will have to become a group effort, and we'll have to involve local organizations and individuals in continuing the follow-up. Especially in the burn zones is where it's most critical, obviously, because this is sort of a one of a kind opportunity to study fire in old growth redwoods. There's not too many places in the state where large chunks of redwood forest have burned hot. And there's even fewer, I would say none, where there is baseline data about fungal diversity and abundance before the fire. And I didn't mean to set up a fire study. By bad luck, some of my transects burned, and now they're available for that. But that wasn't my intention. So it totally changed under me.
Yeah. And I remember you saying in another talk that one of the issues with continuing to do these transects and like pass it along is that it does require the ability to identify what those mushrooms are. It's one thing to do the walking for a kilometer, but it's another to know what you're seeing. But there are ways for us to make it happen.
Yeah. One of the potential ways to solve that issue, which is a good point, is that you can focus on a smaller number of species that are easier to identify. Just limit it to a couple dozen species that even beginners can recognize, so that at least you're capturing the dynamics of the different year, even if you're not getting all the data about the diversity. So there are still bits of data you can get, even if you're not an identification expert.
Yeah. Well, and I'll just say, I mentioned briefly before you started giving your presentation that the museum is launching an initiative in partnership with the Norris Center and with the California Native Plant Society. And it really is based on giving people the resources that they need to go out and make useful observations. And it's starting with kind of more of a plant focus, but we hope to broaden it. And so maybe we can add a fungi side to it in time for next winter. But yeah, definitely, this is an important time for people to be going out there and making observations and adding them to things like iNaturalist. I had a question about your use of iNaturalist so you're using, what was that?
I said never heard of it.
Never, yeah. Well, let me tell you about it. So you have your little notebook and you create your data for for this grant and to report back on that. But is every single observation also going into iNaturalist? Because you also pulled up that like data visualization of your observations. So are your transect observations all also on iNaturalist?
Absolutely not.
Okay.
And there is a lot of reasons for that. One is that it's too much data. If I was making an observation for every single species and its abundance, I would never do it. I could never catch up to it. Two, iNaturalist is a point-centered database. So its fundamental unit of observation is a species in a place on a date, a point. What I'm doing is surveying an area and quantifying my effort and time, or sort of the area and the time, which equals effort, and then quantifying abundance, which is a fundamentally different approach than what iNaturalist uses. In fact, it's much more akin to what the eBird project uses. If you ever submitted a checklist of birds on eBird, that's actually what I modeled it after. And I was really grateful to have that experience with the birding community that sort of basically gave me a ready-made framework for how to collect this data. So I do make iNaturalist observations while I'm out there, but it's not a one-to-one correspondence with the records that I'm making. I would really love if at some point some computer science students or app developers were able to build a platform that made data collection streamlined so I didn't have to transcribe my notebooks, because that takes a lot of time and there's room for error. There's a lot of data checking that has to happen. If I was able to sort of immediately plug in the data in the field that then sort of populated iNaturalist with the right kinds of effort quantified metadata, then that would be ideal. But obviously I'm not the person to build that. Someone else hopefully will do that.
Yeah. Let's see, there's so many things coming in. Okay, so one person, I'm just gonna respond to Anita. Yes, this is recorded. It will be made available tomorrow. I will make that happen. And I'll also send out a follow-up email that will have like a link to that. I'll send it out tomorrow. And then also she's curious about public access to burned areas because she's interested in seeing some of these mushrooms that you have been talking about in this presentation while they're maybe still around. And do you have any advice for that?
Yeah, access to a lot of the burned areas I had to negotiate based on my standing permits. And actually I got kicked back out again while they renegotiated researcher access. So even I don't have unrestricted access to my own transects. And a lot of the park areas are closed. There are a lot of roadside areas that are accessible to the public, especially, Jameson Creek Road, Highway 236, Highway 9, Alba Road, Felton Empire. And it's a little bit dangerous out there, so I really can't recommend that everyone spends a lot of time in burned areas. There's trees and branches coming down. The soil is unstable in some places. So I don't recommend that you flock out there, but if you're interested, you can definitely find roadside areas in the Santa Cruz Mountains that will show some of these fungi right next to the road in publicly accessible areas. But please, yeah, respect state park closures and don't go traipsing around in there.
Yeah, there is one public land area that I know of that is open. I don't know if you're likely to see any of these mushrooms there though, but it's the Bonnie Dune Ecological Reserve, which is the Santa Cruz Sandhills habitat. So not ideal for seeing these like redwood habitat.
Some of the species I mentioned have been found in the Bonnie Dune burn this year.
Yeah, well, like at the Ecological Reserve?
Yep.
So I think the trails that go through the redwoods are currently closed, I think. So it'd be interesting, but we should know. So you guys should all go there and take pictures and upload them to iNat, and then we'll know.
And there's going to be some cool plant life there for sure.
Yeah, I'm excited. That's so for our fire project that we're doing, we are hoping to have some in-person, safe, distanced, masked trainings at the Ecological Reserve, focusing on some rare plants that are there, which there are rare plants.
Find that penstemon. That's the one I want to see.
Yes.
I've seen it before, but only literally like one plant, one little sapling.
Was it like up at like Eagle Rock area?
No, it was actually really close to the Bonnie Dune Ecological Reserve. It's a place where fire management had caused some landowners to mow an area and sort of mimic the disturbance of a fire, and they sprung up in this mode area, but then they declined in the following 2 or 3 years. They just went back down to almost no population.
That's interesting.
So I'm hoping that the fire will stimulate them.
Yeah. Okay, we've got some other questions that are specifically like about the fire, like impact. Let me see. Well, someone's wondering if we're going to see some morels around Santa Cruz County soon. What do you think?
Undoubtedly the morels will be there, and it will be an issue. It'll be a problem because morels are a commercially valuable, commercially harvested species or group, genus, and people will come in large numbers to places to harvest them, and those soils are really sensitive in the years immediately following a fire, and I expect it to be an issue, which I'm not super excited to see how it pans out, because it didn't, there were issues with it after the Bonnie Dune fire in 2008, where there were rangers chasing people around, and it's never very much fun when it becomes that pressurized. So yes, the morels will certainly be there, and so will the people.
Yeah, that kind of brings us to our next question, which is more just like not fire specific, but for your transects, did you have any issues with mushroom hunters disturbing your transects?
Yeah, and I knew that getting into it. I don't own these transects. They're not, I can't ask people to sort of leave them alone. They're public land. And in general, there was astonishingly little disturbance that I could detect. In most cases, I was able to follow fruit bodies week to week and could tell that there wasn't really much going on. There was a few cases, though, where I think 2 or 3 days total, where I was able to detect that there had been harvesting of particular edible species. And I tried to note that in my data, and I tried to estimate based on, because usually harvesters leave behind traces. They don't totally completely hide their traces. So I was able to mention, at least in the notes for those days, an estimate of what I was missing. But because my sites were situated in redwood forest, this is not the ideal habitat for most of the highly prized edible mushrooms in our county. So I had less trouble with disturbance than I expected. It was pretty clear that there was not a lot of removal or disturbance going on.
Okay, cool. So another question about the fire. John says that some plants get a boost from smoke and ash, if not directly in a burn zone. So kind of like residual impacts of fire. Do you have any thoughts on that when it comes to fungi?
Totally awesome question. I feel anecdotally, I haven't crunched the numbers yet, that there were fungi that were not in the burn zone. So in other words, like over at Nicene Marks, pretty far away from where the CZU fire scar was, that had an increase in abundance this year, both there and in the burn scar, which suggests that perhaps there are longer distance chemical signals being, like smoke particles, for example, is a great example, that perhaps, this would be totally amazing to find evidence for, is if there were smoke stimulated fungal fruitings. But I don't know. We'll try and deduce stories like that from the data if they're present.
Yeah, so many questions, which just means that like all the more reason to get out there and start looking. So from Matt, is it possible that mushrooms that have an ectomycorrhizal relationship with certain trees that are fire sensitive or fire intolerant are also fire sensitive intolerant?
That's an awesome question. And it's so sort of elegant and simple, that connection. It's like, if the fungus is totally dependent on its plant host, and that plant host can't survive a fire, then you definitely can predict that you will lose the fungus with it. And that should be the case for all of the Douglas fir associated species. We expect that there will be a major decline in the abundance of the mycelium and the abundance of fruiting of things like slippery jacks, the swillus mushrooms, which are obligate associates of Douglas fir in many cases. So not all slippery jacks, but there are some really common ones in the county that only grow with Douglas fir, and we expect that their populations took a major hit in those areas.
Right, yeah, because if the tree dies, then the mycelium that's associated with it would die. But I guess there's other redwood. Are there redwood associates that you're curious about?
There are redwood associates, but not mycorrhizal associates, because redwood don't form ectomycorrhizae.
Yeah, as I was saying, I was like, wait a minute, I don't actually think that's true. That's interesting. Okay. One thing, all right, so this is from Natalie. One thing I've noticed around where I live, Smith Grade, Bonnie Dune, is that there are almost no mycorrhizal fungi fruiting in the burn zone nearby, even though the fire was not as intense as in your photos and almost all of the burned trees are still alive. Is this due to damage to the trees, damage to the mycelium in the soil, both unknown? So that's kind of right off of what we were just talking about.
I think all of those are correct. It's probably somewhat due to damage to the trees. So if they lose a bunch of leaves, they can't photosynthesize as much, which means that the main product they're providing, or the main resource they're providing to their mycorrhizal partner, which is sugar, photosynthetic product, is dramatically lessened. So perhaps it's the case that the fungus doesn't have the same carbon pool to tap into to produce fruit bodies. So that makes a lot of sense. It's possible that the mycelium itself was affected by the heat of being burned, so that sort of organic matter layer burning above them. That makes a good sort of hypothesis that we could examine. And then in some cases, it's unknown exactly why it might happen. Maybe they'll rebound like crazy next year, and that would be evidence that the mycelium did persist and they just didn't have quite enough sugar. But all of those are plausible explanations, and it probably varies by species. So that will require a much closer look that I don't know. We'll have to see what the data looks like.
Okay, let's see. Does rain seeping through wood ash form something like lye and change the pH of the top of the soil? What would that impact fungi fruiting? So like a chemical soil?
Absolutely, 100%. One of the major impacts on the soil is the immediate basification. So it becomes very alkaline as water leaches through carbon. There's a major flux in the pH of the soil, and mushrooms are super sensitive to pH. In fact, in Europe, it's oftentimes the first couplet in identification keys is, did you find your mushroom in an acidic forest or a basic forest? Which is not something we tend to think about in America, but it's totally the case. Yeah, a lot of these fire-adapted fungi are fungi that likely have adaptations for alkaline soils.
Yeah, so as opposed to like maybe a Santa Cruz cypress that it's like, you know, like heat or smoke that's like opening up a cone, like that kind of reaction, but it's actually like a chemical stimuli that's
to spore germination potentially. Yeah.
Okay, sorry, it's like 6:13. If you gotta go, I understand, but maybe we can just get to like one other question that I see popping up, which is from Lou. It says, could smoke, particle matter, and fire updraft act as an accelerating vehicle for spores released during the fires? In other words, spores are carried much longer distances by the smoke.
I don't know a lot about this, but I did read a couple articles in the immediate aftermath of the California wildfires this year, where biologists who do study this more closely were definitely commenting in favor of this idea that, or in support of the idea that the sort of weather, the pyro weather, there's like local weather that fires create when they burn hot enough and intensely enough, where updrafts are made, and they even make their own clouds, the pyrocumulus clouds. And it makes complete sense to believe that some microbes that have resistant resting stage spores can be sort of entrained in those air currents and moved in a way that they wouldn't otherwise have been moved had not the fire and its sort of associated wind currents come through. But I can't begin to comment on that.
Yeah, and that just seems like such a hard thing for us to be able to know for sure too. Such a hard thing to track. But I want to come back to the fact that like what you have been outlining for us today, though, with your transects is like you're purposefully doing it to be something that's repeatable by other people and like and manageable. And so maybe could you sort of like leave us with a parting thought about how you'd like us to take on your work or what you'd recommend?
Let me see if I can get this. I'm going to drop a link in the chat, I hope.
Yeah, and I've been dropping links too. So while you're doing that, there's a link to purchase a copy of Christian's book, Mushrooms of the Redwood Coast, or MORC. And then also a link to support the Norris Center. the link to support the San Lorenzo Valley Museum. I'm gonna add some more links, but also Christian's adding links. So what do we got here?
So this is just a really brief write-up I wrote about what the long-term monitoring protocol is and why the various steps exist the way they do. And basically the idea would be for you to either, for people, for the community in Santa Cruz to follow up on my transects that I can provide GPS sort of tracks for, or to establish new ones on your own and set up a sampling schedule that you implement this protocol, this basic protocol on. And yeah, just be in touch with me. I'm easy to find, and you can ask me questions about how to do this. And it takes time to learn it. You have to sort of get in a groove of how to go out and record this. But I did try and make it as easy as possible. This is not something where you have to do really rigorous, super arcane ecological study methods where you like randomize numbers and put out quadrats and do a lot of complicated data collection. Just to clarify, someone just asked, how long does it take me to walk a transect? A 1 kilometer transect, when it's not super mushroomy at the beginning of the season in October, takes me half an hour or so, let's say, at the fastest. And in the middle of mushroom season, when it's cranking, it might take me 2 hours on a really diverse, really wild transect. But usually it's somewhere in the vicinity of an hour. I would say the average is 45 to, let's say an hour. Probably an hour is the average.
Yeah, I'm inspired to try to get something going next winter where we can help facilitate something like this, because I want to do it myself anyways. I'm also going to be sending out, like I said, a follow-up email, and I will include this link in that, some other resources, and again, lots of links, lots of other links I've put in here. There's a link to keep in touch with us. So if you want to find out about future events like this, you can join the museum's newsletter list. We send out periodic emails. And then also there's a link to a survey. We'd love to hear what you thought about this experience. And I'll send that in my follow-up email as well. And it really, really helps us out. So it just takes a moment for you guys to do that. So please consider filling out that survey either right now or when I send it to you tomorrow, along with a link to a recording for this. I want to again thank Chris Lay from the Norris Center. Chris, are you still here? You can like pop on in and say any parting words. But yeah, definitely thankful to the Norris Center and to the San Lorenzo Valley Museum. And please, I'll also put in here a link to the exhibit. Chris, I see that you're back.
There I am. Hi, thanks everybody. This was fun.
Great, yeah. So there's a link to the exhibit, Look, Act, Inspire. And then also there you can find the link to the next event in this series, which is about Fred Mcpherson, which is really exciting. So yeah, lots to come, lots going on always. And thanks so much for joining us tonight. And Christian, thanks as always. Can't wait to have you back.
Thanks for having me. Yeah.
Yeah, thanks, Christian.
All right. And Christian's also, I don't know if we mentioned this, but Christian's featured in the Look, Act, Inspire exhibit as one of the featured naturalists. So look for that. Okay. We will leave it at that. Go enjoy your dinner. Good night.
Bye, everybody.
Bye, everybody.